Hello class and welcome to lab 4. This is on muscle physiology. I'd encourage you guys to read through the lab in your lab manuals, and there's a few questions to answer that I'll help explain before you get to the lab report questions. But again, you can answer those lab report questions in a separate document, or you can write them down and take a picture of them when you submit them for the assignment. So when we talk about muscle physiology, in lab we would perform probably a computerized demonstration, or we would use some sort of simulation software to look at stimulating a muscle and then measuring the force of contraction. There's a great YouTube link that I also included under lab 4, and it just shows kind of an example of a computerized software system of performing some of these experiments. So what is a motor unit, first of all? A motor unit is a single motor neuron and all the muscle fibers it innervates. All these muscle fibers in a motor unit will contract at once. So a motor unit, again, is a single motor neuron and all the muscle fibers it will innervate. Graded contractions are varied contraction strengths due to different numbers of motor units being stimulated. The neuromuscular junction is the site where a motor neuron will stimulate a muscle fiber, and the motor end plate is the area of the muscle fiber, sarcolemma, or kind of the membrane of that muscle fiber, where a motor neuron will stimulate it using the neurotransmitter acetylcholine. So this is a look at the anatomy of a neuromuscular junction. The name literally means junction between the neuron and between the muscle. And also the motor end plate. And the motor end plate is again on the muscle fiber that each motor neuron innervates. So if we zoom in here, we see synaptic vesicles, which are little tiny dots that contain the acetylcholine, the transmitter, that will be released and cross over the synaptic cleft onto the motor end plate when a muscle fiber is stimulated. And here you can see the neuromuscular junction, which shows the somatic motor neurons, so the nerve endings. And the neuromuscular junction is just where those nerve endings are connecting to the skeletal muscle fibers that it will innervate and stimulate. So the motor unit, again, definition of it, it's the one single motor neuron and all of the skeletal muscle fibers it will innervate. So it'll have neuromuscular junctions, so it'll have different branches off of each motor axon, and each will end in a neuromuscular junction, which will release that acetylcholine, that neurotransmitter, to innervate the skeletal muscle fiber. And we'll go over this more when we get to the muscle system and all the physiology behind it. But we're more talking about control of these motor units and stimulating the muscles to measure their contraction and strength. The control of motor units, contraction strength comes from motor unit recruitment, which just means recruiting more neurons to stimulate more muscle fibers, really for strength purposes. Finer muscle control requires smaller motor units or fewer muscle fibers. So for example, your eye muscles, your other small muscles in the body, your finger muscles, they might have about 23 muscle fibers per motor unit. Larger, stronger muscles, like your quadriceps muscles, may have motor units with thousands of muscle fibers. Control and strength are trade-offs, and what that means is that your eye muscles, your finger muscles are extremely precise, so they have very precise control in terms of how you can move them and control which direction your eyeball sees in lots of different directions. So that's control. But strength, the strongest muscles of the body are usually the larger muscles. They'll have more motor units, but they'll have less control. You can only move your leg really in two directions, extension and flexion. The study of skeletal muscle contraction is done in vitro in lab, where one end of a muscle will be fixed and the other is movable. And a lot of times we use stimulations if we don't have a muscle or an animal muscle to use in lab. Electrical stimulations will be applied to this muscle, again, where one end is fixed and the other is movable. And then we can record the contractions by their strength and display them as currents, as electrical currents that go through them. So a twitch is when a muscle quickly contracts and relaxes after a single electrical shock of sufficient voltage is applied to the muscle. Increasing the voltage will increase the strength of the twitch up to a certain maximum. So muscle cells do not contract or have kind of a unending amount of strength. There is a maximum strength that the skeletal muscles will have. When a second shock is applied immediately after a first shock, the second twitch might partially piggyback off the first, and this is called summation. And I'll show you a picture of what I mean by that. The latent period is just the time between stimuli and the contraction. And this is the time when the myosin and actin will form a cross bridge with each other. And the myosin and actin are myofilaments that form cross bridges to help your muscle fibers to contract. Graded contractions are stronger contractions resulting in recruitment of more fibers until all fibers are contracting at once. So this is a look at a single stimulus or electrical shock to a muscle and showing how the muscle will twitch or respond to that electrical shock. Summation, if you put a second stimulus of equal strength really close to the first stimulus, these twitches in the muscle might piggyback off each other. And this is what we call summation. So it would actually form a larger muscle twitch in kind of the second, after the second stimuli, because they will piggyback off each other. Tetanus is increasing the frequency of electrical shocks. And when we do that, that decreases the relaxation time between twitches. This is called incomplete tetanus, when there's still an amount of relaxation time between twitches. At a certain frequency, there will be no relaxation at all, and this is called complete tetanus, which will be a smooth, sustained contraction where your muscle is constantly contracting. It cannot do this forever. We'll see what happens. So incomplete tetanus, again, 5 shocks per second. This graph is showing 10 shocks per second. Again, these are electrical voltages that are being applied to the muscle, showing how the muscle tension changes between as we move up in our shocks per second. There's still some relaxation time between twitches. Even in the 10 shocks per second, you kind of see a jagged line. So that shows that the muscle is relaxing a bit between shocks. So that's why we call this incomplete tetanus. And complete tetanus is when we have one sustained contraction. So it's a nice solid parallel line. You won't see any kind of relaxations between those shocks. And eventually the muscle will start to fatigue. You cannot completely electrically shock your muscles forever. The muscle tension will eventually decrease and the muscles will start to fatigue or break down. Tetanus in vivo is the asynchronous activation of motor units, where some motor units will start to twitch when others start to relax. This will produce, like we talked about, a continuous contraction of the whole muscle, and recruitment will make the contraction stronger. Treppe or Treppe is kind of a staircase-like effect. If we increase the voltage, the number of muscle fibers used in vitro in lab increases. This will reach a maximum value when all muscle fibers are stimulated at once. If a fresh muscle is stimulated with several shocks at this maximum voltage, each twitch will become progressively stronger. And we call this kind of a staircase-like strength in muscle strength increases, Treppe or Treppe. So think staircase is the Treppe effect or Treppe effect. Muscle tone is the maintenance of partial contraction of a muscle, and this is important for generating reflexes, maintaining posture and balance, and controlling proper function of other organ systems. So a lot of your smooth muscles in your intestinal tract are under partial contraction as they're constantly moving waste and food products along your digestive tract. Also, the muscles in your back are partially contracted at all times to help maintain posture, to help you sit up and stand. Tone is controlled by the sensory muscle spindle, which measures muscle stretch. And this is just a good example of describing muscle strength versus muscle tone. Muscle strength is defined as the muscle's ability to contract and create force in response to resistance. And muscle tone is defined as the tension in a muscle at rest. So now we get to muscle twitch, and these slides are just kind of another kind of general review of what we talked about, but maybe a different way of explaining it. A muscle twitch, again, is an involuntary contraction of the fibers that make up a muscle. Nerves control muscle fibers, and when something stimulates or damages a nerve, it causes the muscle fibers to contract, resulting in this twitch. When we're not in lab, you could actually see a twitch. Most likely, you can often see it below your skin as your muscle contracts. These are 3 phases of a muscle twitch: the latent period, the contraction period, and the relaxation period. And again, the single muscle twitch has 3 components. The latent period is the lag phase, the contraction period, and the relaxation phase. This kind of describes in depth the 3 different phases and periods. And you guys are free to pause the PowerPoint at this time to read through and a little more explanation of those 3 periods of the muscle twitch. That might also help you answer a lab report question. Muscle responses to changes in stimulus frequency. We talked about summation occurs as successive stimuli are added together to produce a stronger muscle contraction, and tetanus is the fusion of contractions to produce continuous contraction. And here again is a great graph showing how a twitch, one stimuli will produce one twitch, and if the stimuli are successive right on top of each other, the twitches might summate or piggyback off each other. Incomplete tetanus shows many stimuli, but we still get a relaxation period between the twitches themselves, so we call that incomplete tetanus, whereas fused or complete tetanus shows a constant contraction state of that muscle with constant stimuli. Muscle responses to changes in strength. Motor unit recruitment refers to the activation of additional motor units to accomplish an increase in contractile strength in a muscle. So it's just recruiting more motor units to increase the contraction strength of a muscle. And this, again, just shows stimuli to a nerve and showing how the nerve fibers being excited in that muscle fiber will be more recruited or kind of called upon to increase the maximum contraction strength. You'll see here that regardless of the stimulus voltage, after a certain amount of time, and specifically, what is it, around 7, 8, 9, even though the stimulus voltage will increase, so you're sending a greater electrical shock to the muscle fiber, because all the nerve fibers have been recruited and excited, there's only a maximum contraction strength or tension that each muscle fiber has, because there won't be any other muscle fibers to recruit to make it stronger. And that's the end of this PowerPoint slide. Put any of your questions, post them in the discussion for this lab, and I'll try to be checking my computer throughout the week to answer your questions.